हाँ जी हाँ जी। लगा लीजिए।
Very good morning to all. So today is your last lecture on parasitology, and we are going to talk about entomology. As you all know, parasitology is studied under three heads. One is protozoa, the other one is helminths, and the third part remains the arthropods, which come under entomology. So in today's class, we should first understand what is the meaning of the term medical entomology. Now basically it refers to the study of diseases which are caused by arthropods. To make an understanding of the arthropods, how they affect us in different ways, arthropods affect the health and well-being of humans and animals in multiple ways. This includes they can act as direct causes of disease or distress to humans. They can act as vectors or host of pathogenic organisms, or sometimes they may be helpful as they act as natural enemies of other medically harmful insects. So arthropods play roles in our lives in various ways. They may be harmful in many times, but they have many beneficial effects as well. So they can act as direct causes of disease or distress, and I will give some examples for that. They can be ectoparasites such as ticks, fleas and mites. They may act as endoparasites. A condition example is myiasis, which is caused by the maggots of the flies. Then there is a condition known as envenomization, which may be seen with some arthropods like wasps, bees and spiders. And they can also lead to allergic reactions, as happens in, for example, in case of dust mites. They can also act as vectors for various pathogenic organisms. The vector basically refers to living organisms that can transmit infectious diseases between humans or from animals to humans. We will see the details in the subsequent slides. They can also act as natural enemies of other medically harmful insects. For example, mites may be parasitic on mosquitoes, and fire ants consume the tick eggs. So they can be beneficial to us by destroying the other arthropods or other insects where these insects may be harmful to humans. Now, the phylum Arthropoda, which actually covers most of the things we are going to study today, it includes lobsters, crabs, shrimp, centipedes, millipedes, insects, ticks, mites, and spiders. So there is a wide variety of arthropods which we can see around us, but some of them are of particular concern for us in terms of health and disease. There are over 1 million species of arthropods, and it makes up the largest phylum in the animal kingdom. Coming to some general features of Arthropoda, all of the arthropods are bilaterally symmetrical. They have jointed legs, as the term suggests, arthropods, so they have jointed legs. They have a dorsal heart, which is an open circulatory system. There is a central nervous system which is well organized, and there are striated muscles in all of them. Arthropods show metamerism, they are metameric. This means their body exhibits true segmentation, that is, they have individual segments showing a set of muscles and nerves, so they are metameric. They show tegmatosis. This means their segments may be modified in a way that they are grouped together to form mouth parts and other body structures such as thorax. All the arthropods also have a chitinous exoskeleton, which is a nitrogenous polysaccharide which helps it to tide over the adverse environmental conditions. Now, the medically important arthropods can be classified into various classes. The most important class among these is Insecta, and there are different orders. For example, Diptera contains many of the important vectors such as mosquitoes, sandflies, black flies and tsetse fly. Then there are some common other insects such as houseflies, flesh flies, biting midges. Again, they all are covered under order Diptera of class Insecta. Then there is another order which is Siphanoptera, where the fleas belong to. Then there are two more orders, Anoplura and Malophaga, where you will find the lice. So these are some of the important orders under the class Insecta which we will be dealing today. There are other classes as well. The other important class for humans is Arachnida. So first was Insecta, now we have Arachnida, where you will find hard ticks, soft ticks, mites. All these belong to the class Arachnida. We will see what are the differences between Insecta and Arachnida. And the third one is Crustacea. The crustaceans, basically the most important crustacean for us which has a role in the transmission of diseases is Cyclops, and the other one is Diaptomys. Then there may be some other crustaceans such as crab and crayfish which may act as intermediate hosts in the life cycle of some of the parasites. So these are the major classes and orders where most of the most important arthropods for humans belong to. Now, how will you differentiate them? There are some things which is common to Insecta, all Insecta, and there are other things which are common to crustaceans and Arachnida. We will see how they are different. First, we will start with some examples of Insecta. This include mosquitoes, flies, lice, fleas and red-winged bug. Arachnids include ticks and mites, and crustaceans include cyclops. When you see their body divisions, in case of Insecta, they have their body divided into head, thorax and abdomen, while in the other two, that is Arachnida and Crustacea, the body divisions are cephalothorax and abdomen. So head and thorax are fused to form cephalothorax, and the second segment will be abdomen in both of these classes. Now there are 3 pairs of legs in Insecta, while arachnids show 4 pairs of legs and crustaceans have 5 pairs of legs. Now these features will help you in differentiating whenever you get any specimen where you have to differentiate the different insects or arachnids. These features will help you in determining which class this particular specimen belongs to. You can also look for the antennae. In Insecta there is only 1 pair of antennae, while in case of Arachnida there are no antennae present. In case of crustaceans, 2 pairs of antennae are seen. Insecta can have a single or 2 pairs of wings, or they may be wingless as well, while arachnids are all wingless and crustaceans also are wingless. So if you find any arthropod which has wings, it belongs to Insecta only, while wingless can belong to any of the 3. Coming to our first major group of arthropods today, that is mosquitoes. Mosquitoes belong to family Culicidae with more than 3200 species and subspecies, with 37 genera reported worldwide. Mosquitoes and ticks together account for the majority of transmissions of the most important vector-borne diseases in human beings. And as we have seen, they are insects, so their body can be divided into head, thorax and abdomen. There is 1 pair of 4 wings, and there are halters or hind wings which are vestigial in mosquitoes. They have compound eyes. There is a proboscis which is a piercing mouth part, and then there is a pair of antennae. The family Culicidae can be further divided into 2 subfamilies. First is the Anophelinae and the other one is Culicinae. And under the Anophelinae we have 1 genus of importance, that is Anopheles, while Culicinae we have 3 genera, Culex, Aedes and Mansonia, which have which act as vectors for different diseases. Now coming to some differences, the important difference between the two subfamilies, anopheline and culisine, we find that first we need to differentiate the eggs. For example, in case of anopheline eggs, you will find they are again boat-shaped looking eggs, but they have floaters on the sides. While in case of culisine, which includes both the Aedes and Culex, there you won't find any floaters or floats like this. In case of culisine, they don't have any float, but they can be recognized based on their arrangement. In case of Aedes, you will find individual eggs floating just beneath the surface of water, while in case of Culex, you will find these eggs are arranged in the form of a raft floating on the top of the water surface. Coming to the larval stage, anopheline mosquitoes larvae do not have any siphon tube. Now this is a tubular structure, as you can see here. This is a tube that helps these larvae to derive oxygen or take oxygen from the environment, from the outside air. Since they are there inside the water, this siphon tube, they hang from their siphon tubes, and this siphon tube takes up the air from the top air, and they can take in air this way. While in case of anopheline larvae, since there is no siphon tube, they float parallel to the surface, and you will find them floating almost parallel to the surface in where they are breeding. In case of culisine mosquitoes, they can hang almost vertically to the water surface, and you will notice a siphon tube which is there, which can vary depending on the species. So this siphon tube is helpful in differentiating culisine from anopheline larvae. The third stage is the pupa stage, which is almost similar in both the subfamilies, so I won't go into much detail of these two. Coming to some more specific details about these mosquitoes, as we have seen, the siphon tube or the breathing tube is rudimentary or almost negligibly present in case of Anopheles, and they lie parallel to the water surface. In case of Culex larvae, as you can see, there is a siphon tube, but this siphon tube in Culex is short and stout. While in case of Aedes, this siphon tube is long and slender. Also, you will find there is only a single pair of hair tufts around this area, while there are several pairs of hair tufts surrounding this area in case of Aedes mosquitoes. Pupae are similar. In case of adult anopheline mosquitoes, when they sit, you will find that their proboscis and their body, they are almost in straight line. In case of Culex and Aedes, they lie at an angle. The proboscis and the body are at an angle. So this is how you can again differentiate their different stages of different subfamilies. An additional feature is that when you see an anopheline mosquito, you will find that their wings have spots. These spotted wings will help you in differentiating a mosquito, adult mosquito of Anopheles. While in case of Culex, you won't find any special structures like this. While in Aedes, you will find their body and legs are striped. So they look like, again, because they are also commonly known as tiger mosquitoes, so they have striped body and striped legs. So this is how you can identify the Aedes mosquitoes. Coming to the life cycle, all the mosquitoes have a simple life cycle starting from the eggs which are laid on near the water surface. Thereafter, there is a development of a larva which subsequently transforms into a pupa, and eventually an adult mosquito is developed, which emerges from the water surface. Now we will talk about something about the behavior of the mosquitoes. So how their behavior can have effect on your life. So the female mosquitoes are the main are the ones which actually are going to feed on animals and humans, and they are responsible for transmission of the disease agents from human to human or from animals to human. Now mosquitoes are attracted to a host by the way of their body odors, carbon dioxide, and also heat which is emitted from animal or person. Some species have a habit of biting during certain hours. For example, some of the species prefer biting during dusk time, some prefer biting during dawn time, some of these may prefer biting in the middle of night. So there are different habits for different species. Most of the times mosquitoes feeding usually takes place during the night, but daytime biting can also happen, as you may see in case of Aedes. Some species prefer to feed in forest areas, while some prefer biting outside of houses, or others may prefer biting within the houses. So all the mosquitoes have different habits, and accordingly, if there's knowledge about these things, maybe you can take appropriate preventive and control measures depending on the type of species which is prevalent in a particular geographical location. Now coming to some of the common mosquito borne diseases and their vectors, amongst the viruses, Japanese encephalitis is one virus which is transmitted by different species of culex, the most common being culex tritineorhynchus, and there are other species as well. Then dengue virus or chikungunya virus or even Zika virus, the main vector remains Aedes mosquito, and there are two main species, Aedes aegypti and Aedes albopictus, which are involved in transmission. When we talk about the protozoa and nematodes, malaria, which is caused by various species of Plasmodium, the vector here is the female Anopheles mosquito. Again, this female Anopheles species is going to transmit malaria, while filariasis caused by Wuchereria bancrofti is transmitted mainly by Culex species, and the other one is Anopheles species. While Bruguier which is another important agent for lymphatic filariasis, is transmitted by Mansonia. It can also be transmitted by Aedes toboi and Anopheles sinensis. So there are multiple species which may help in the transmission of Bruguier malaria. Both these agents can present with lymphatic filariasis with varying severity. Coming to flies now, the most common fly you see is housefly, and housefly can help in the transmission of disease by different ways. It can act as a mechanical vector where it can carry viruses such as hepatitis virus and poliomyelitis virus on their legs or wings, or they can also help in transmission of bacterial agents, for example Salmonella leading to enteric fever, bacillary dysentery, wound infections and trachoma. So here they can act as mechanical carriers where the agent may be carried on their wings and feet, and when they sit on from one area to another, they can carry these agents to a susceptible person. They can also carry some parasites. For example, they can act as a mechanical transmission agent for eggs of Ascaris lumbricoides, Trichuris trichiura and Hymenea lepis nana. They can also act as transmission agents for cysts of Entamoeba histolytica, Giardia ramblia and others. So they can act as mechanical vectors for many diseases. They can also be responsible for a condition known as myiasis, which basically refers to invasion of living tissues of man by dipterous larvae. So they are larvae of these flies. They may invade the living tissues of man, and this condition is known as myiasis. There are different clinical types of myiasis which have been described depending upon the site which is involved. It can be a cutaneous myiasis where skin is involved. It could be intestinal. It could be atrial, which means a cavity is involved, such as oral cavity, nasal, oral, ocular, sinusal, vaginal and urethral. So various cavities may be involved by these larvae. And then they can also infest the wounds, and this condition can also happen in and this is more common in people who are physically debilitated and who cannot take care of themselves or who have very poor hygiene. In those situations it is more likely to happen. The next group of flies is belongs to the family Phlebotomini, and it is again looking like a mosquito only, but it is not a mosquito, it's a fly. It is very similar to a mosquito, but its size is a little smaller. Its body is very hairy and the wings are feathery. As you can see, there are a lot of hair on the body of these flies. These are tiny insects as compared to mosquitoes. They are just around 1 to 3 millimeter of size, and they have a lot of hair all over their body and wings. And the wings rest over their body as if they are angels' wings. So they sit over the body just like as if they look like angels' wings here. Again, like mosquitoes, it's only the females that suck blood, and they are responsible for transmission of the agents. The most important member of this family is Phlebotomous species, particularly Phlebotomous argentites, and we commonly call it sandfly. These sandflies live in warm climates. They are minute and hairy as with other Phlebotomini, and their wings are kept erect while resting. These sandflies breed in the cracks of soil and wall, or also in rubbish heaps. They don't fly, they don't fly much, they basically hop. So they are not going to reach higher floors. In case there are some sandflies, they are going to stay low, they are going to hop only. They have a nocturnal biting habit, and during the daytime they will rest in cool and damp places. Their significance lies in the fact that they are the vectors for leishmaniasis, also commonly known as kala-azar. So visceral leishmaniasis or kala-azar, and also for cutaneous leishmaniasis and other presentations, they act as vectors. They can also lead to sandfly fever. So these are important vectors in that scenario, because kala-azar is an important public health problem for India, particularly for some states like Bihar and Eastern UP, where they are found more commonly. Based on these features, you can identify sandfly as we have discussed in the previous slide. Another important feature to see is in the wings of these sandflies. When you see the wings closely, you will find the second longitudinal vein, this one, divides twice. The first division, then there is a second division. So this 2 times dividing longitudinal veins is again a feature which will help you in confirming that it is a phlebotomous species. Another important fly is CC fly, which is responsible for the transmission of African sleeping sickness, and the agent is Trypanosoma species. So this is another vector, but this is not seen in India. It's mainly a problem seen in African countries. Now coming to our next group of arthropods, which are fleas. We'll talk about the fleas of medical importance here. Xenophila chiopis is the most important flea you know of, and they are the main vectors for plague, particularly bubonic plague. When you see these fleas, they are wingless. They are insects, yes, they are insecta, but then they don't have any wings, and their body is flattened side to side. So they are flattened side to side. So by seeing such a structure and body divided into head, thorax and abdomen, 3 pairs of legs, all these features, and with the side to side flattened body, you will easily identify a flea. So what are the things which can be transmitted by a flea? The most important one being bubonic plague. They can also act as vectors for endemic or murine typhus, or even for helminth, which is Hymenolapis diminuta. The mode of transmission for all these things could be either biting. At the time of biting, they can introduce the infectious agent. It could be a mechanical transmission, or it could be even a fecal transmission, where when they are sitting, when they pass out the feces, that may be carrying the infectious agent, and during scratching of the area, the infectious agent may be introduced into the area where they have sat on. So that is how there are multiple ways they can help in transmission of various infectious agents. The next group of arthropods is human lice, and when we see the lice, they are again insects. They are small wingless insects. They are basically 3 species. Actually, there are 2 major species, and it's a Pediculus humanus, which has 2 subspecies, which are Pediculus capitis and Pediculus corporis. The Pediculus capitis refers to head louse, while body louse is Pediculus corporis. Both these lice look very similar, and most of the times the body louse will be found living in the clothing, and it attaches its eggs to the fibers of the clothing. While in case of the third species, which is human lice, which is also known as Thyrus pubis, or the crab louse or pubic louse. Now in this case, they are mostly found in the area of the pubic hair, or also in the area of the eyelashes or eyebrows. Now when we talk about the head louse, which is Pediculus humanus capitis, it lives in the hair and attaches its eggs or nets to the hair. Its body is dorsoventrally flattened, has head, thorax and abdomen, which is similar to those that seen with other insects. So that is how you can simply identify louse, and louse can be responsible for multiple infections. Before that, we will talk about Thyrus pubis. Now, as compared to head and body louse, which look very similar, Thyrus pubis or pubic louse is quite different. It resembles more like a crab. It has a more quadrate shape. It is not that elongated. It's just around 2 mm in size. It attaches primarily to the pubic hair and eyelashes. Its body, like other insecta, can be divided into head, thorax and abdomen. It has 3 pairs of legs, and first pair of legs, as you can see here, it is very slender. So out of these 3 pairs of legs, the first pair is slender, and the last appendage ends in claws. So these are the features: a quadrate shape, a thin slender first pair of legs, and claws at the end of the last appendages. These features will help you in identifying a pubic louse or thyrus pubis. Also, you will find that the abdomen has lateral projections known as lateral papillae, and in case of females, the last abdomen segment is bifid, while in males it is not seen. So these are the features by which you can even differentiate males and females. Now, what are all the diseases which can be transmitted by lice? Lice can act as vector for multiple conditions, particularly some rickettsial infections such as epidemic typhus, epidemic relapsing fever, and trench fever. Besides that, they can also themselves be responsible for the discomfort to the patient. For example, in a condition known as pediculosis, they stay in the hair region or on the skin, and there they can cause irritation and pruritus. So this is pediculosis. This is another condition which may be caused by lice. The next group of arthropods is ticks. Now ticks are different from insecta. So lice were insecta, while ticks and mites all belong to arachnida. So as you must be remembering from our previous slide or earlier slide, you find that there are a lot of differences between insecta and arachnida, and that you can apply here. These are wingless insects. They are wingless arthropods, basically not insects in the correct way. They are ectoparasites of vertebrate animals, and all of them suck blood. So you will commonly find them if somebody is keeping pets or must have seen some animals. You are likely to see these ectoparasites on these vertebrate animals. They all suck blood. So they are different from insecta in the way that they have 4 pairs of legs here, and their body cannot be differentiated into head, thorax, and abdomen. Instead, they have a cephalothorax zone, and then there is the abdomen. So you don't find it split into head, thorax, and abdomen. Like most other arthropods, males are smaller than females, and in males, body is completely covered by a shell-like structure, which is a chitinous shell known as scutum. This is the shell in case of males. While in case of adult females, it is not covering the entire dorsum, it is only covering the anterior 1/3 of the dorsum. This scutum is different in both males and females. Another important thing to note about the heart ticks is the vertical transmission from mother to egg is possible. So if it is harboring some infectious agent, it is possible that this infectious agent may be transmitted from the mother to the eggs. So this has an important role in continuation of some cycles of infectious diseases in an environment. The other group of ticks is soft ticks. So hard tick was known as hard tick because it has a scutum, which is a chitinous shell on the back side, while soft tick lacks this chitinous shell. They are small, soft, and leathery looking. They have a leathery cuticulum. Their mouth parts are invisible from the dorsum side, unlike the hard tick where you can see from the dorsum, you can appreciate the cephalothorax, and even maybe you can say its head only. So when you see this part, you can appreciate it from the dorsum, while in case of soft ticks, this is lying on the underside. So mouth parts are not visible from the dorsum here, it is on the ventral side. The scutum is absent, so this is the main feature which differentiates soft tick from hard tick. Otherwise, they both have 4 pairs of legs, they are wingless, and all of them are going to suck blood from the vertebrate animals. Now, hard ticks can act as vectors for multiple infectious diseases, including Lyme's disease, babesiosis, tularemia, Queensland fever, Rocky Mountain spotted fever, tick paralysis, and casual forest disease. All of these diseases can be transmitted by a hard tick, while soft ticks can act as a vector for relapsing fever, Q fever, and casual forest disease. Coming to our next arthropod, which is mites, and the most important example amongst mites is Sarcoptes scabiei, which is responsible for scabies. The mites are different from the ticks. One thing is they are very small, they are very tiny. It will be very hard for you to see them by naked eye. They are just around 0.3 to 0.3 millimeters in size, and you generally require some kind of microscopic help to see them properly. In case of Sarcoptes scabiei, which is a scabies causing mite, they are endoparasitic mites. they live in the epidermal layer of the skin. So they live in the epidermal layer of the skin and cause a lot of pathogenesis there, which is responsible for all the manifestations. So Sarcoptes cabii causes human scabies. It is characterized by intensely pruritic, erythematous, papular eruptions which are caused by the adult female which is burrowing inside the epidermis, and it creates serpiginous burrows, and there is a lot of itching, and which is more intense at night. So these are the features which will raise suspicion of scabies, and when you closely examine you will find all these features, and then maybe you can look for the mite inside these lesions. The preferred sites include the interdigital areas, popliteal folds and groin areas. So all these areas are the preferred site for the Sarcoptes cabii. So after the mite, which we talked about the Sarcoptes cabii, we come to our next arthropod, which is a crustacean. The most important example being Cyclops. Now crustaceans can be differentiated from insecta and arachnida. If you remember from our earlier slide, they can be easily differentiated because they have 5 pairs of legs, they have 2 pairs of antennae. And now Cyclops particularly has some interesting features. It has a pear-shaped look, and it has a semi-transparent body. As you can see, it has a semi-transparent body. There are 2 pairs of antennae, and there is a single median cyclopean eye here. So this is these are the features by which you can easily identify a cyclops. They have a large cephalothorax, so there you don't find head and thorax again separate because it's a crustacean. So like arachnida and like crustaceans, all crustaceans, again here you will not find any separate head and thorax. It's a joint cephalothorax, and there is a slender segmented abdomen. Now the cyclops can have a role in the life cycle of many parasites. A classical example is guinea worm disease, where it acts as a vector in the life cycle of Dracunculus medinensis. Now coming to some interesting concepts in vector-borne diseases. Now vector biting activity is one feature which you should understand, because this may have some importance in the public health, because you will need this kind of information for controlling the transmission of some of the vector-borne diseases. For example, some vectors may have a nocturnal biting habits, other may have a daytime biting habit which is diurnal, or some of these vectors may have a crepuscular kind of habits, where they have a biting, they prefer biting during twilight zones. Now the host specificity again can vary in different vectors depending upon the source of blood meal they prefer. They may be amphibophilic, where they like biting the humans. They may be ornithophilic, they like biting more, they feed more on the birds. And they could be zoophilic, where they like biting more of other animals, primarily the vertebrates. Based on their feeding locations, they may have a preference for exophilic or endophilic kind of behavior, whether they like biting outside the houses or inside the houses. So on this basis they could be exophilic or endophilic in nature. Their incubation periods are also useful in many aspects. For example, they can be classified into extrinsic incubation period and the intrinsic incubation period. Extrinsic incubation period refers to the period of incubation which is seen within the arthropod, which is a vector or intermediate host, while the intrinsic incubation period is the one which is seen within the vertebrate host or the definitive host. Now this extrinsic incubation period is generally the time which is required for the infectious agent to develop within the vector to the stage where it becomes infective to the definitive host. Now there are different types of pathogen transmission which are possible, which could be direct, which is spread by direct contact. It could be mechanical, where it is spread by a carrier without any development or multiplication happening inside the vector. Or it could be biological, where there is some multiplication or development within that pathogen which is going to happen inside the vector. So they could be mechanical or biological, or sometimes direct transmission. So when the vector is acting as a mechanical vector, they only act as carriers of these infectious agents on their body surfaces. While in case of biological, there is a multiplication or developmental stages which are seen, as we have seen in the previous slide. So coming to the biological development in more detail, it can be of 3 types. It could be propagative, where only multiplication of the agent is happening in the vector. They are increasing in numbers, but there is no developmental change or cyclical change. For example, in case of rat flea, plague bacilli show such a behavior. So they are going to multiply, but they are not going to show any cyclical change in the body of a rat flea. The other type of biological development could be cyclopropagative, where there is multiplication of the agent inside the body of the vector, but at the same time there is a developmental change as well, which is a cyclical change. And this is highlighted by the main example of malaria, where Plasmodium species, which is using female Anopheles as a vector. Here the Plasmodium not only is increasing in number, but also it is changing stages from trophozoites to schizons to in the Plasmodium it is changing stages from ookinete, sporozoites. So the multiple stages of these Plasmodium may be seen within the body of a female Anopheles. So they are both multiplying as well as showing changes in the body of a female Anopheles mosquito. The third type of biological development is cyclodevelopmental, where only cyclical changes is happening. There is no increase in number of the infectious agent. For example, in case of filarial parasites, for example Butyria bancrofti or Brugia malae, in the mosquitoes there is a development of the parasite so that it reaches the stage where it can be transmitted to a next host, but they are not increasing in number inside the mosquitoes. Similarly, we will find guinea worm embryos. They again undergo developmental changes in cyclops, but they are not going to increase in number inside these vectors. Now, another important point to note is that within the biological transmission, there are some pathways which may be seen, where the biological pathogen transmission, when we are considering, you will also see that sometimes there is a vertical transmission happening. Now, by this we refer to the transmission of the infectious agent to the progeny. So, for example, it's a transoviral transmission seen in Aedes mosquitoes and dengue viruses, where the infectious agent can be transmitted from the mother to the eggs or the developing embryos. Similarly, it is true for Rickettsia rickettsiae in ticks and rickettsial pox which is caused by Rickettsia acari in case of mite. Horizontal transmission basically refers to transstadial transmission, that is transmission of the infectious agent from one stage to another. So it is not transmitting vertically from mother to the eggs, but it is transmitting laterally from one stage to another. For example, in Lyme's disease, in case of ticks, this can happen. In case of rickettsial infection of the spotted fever group, in case of ticks, again transstadial transmission is seen. Also, in case of rickettsial pox caused by Rickettsia acari, in case of mites, this kind of transmission is seen. Now, this is a table which gives a compiled list of the more important vectors and the diseases which may be transmitted by the different vectors. So this is a comprehensive list, and most of these things have been covered in individual lectures of parasites or bacteriology or virology, and most of these vectors have been covered. This is a compiled list which you can use for the reference. Okay, thank you very much. If you have any queries, you can just email it to me, and I will try to get back to you with the answers. Thank you very much. हो गया मैम हाँ ठीक है माइक बंद हो गया ना बंद हो गया डेली हो रही है इसमें क्या डिस्कस होता है